welcome everyone, and especially Constantin, who is our speaker today. So Constantin, like me, studied physics in Heidelberg, and then, unlike me, he did a PhD together with Brett Hambricht and Anna Kreschuk, a little bit at the university in Heidelberg, actually also a little bit at Janelia, and then mostly here at EMBL, and that's actually where I met him, and we also collaborated on some cool projects together, so this was really a great time. Thank you, Constantin, for collaborating with me. It's really improved my life. Very nice. And then he did a very short postdoc, and then immediately became a junior professor in Göttingen, where he is leading a research group with the name Computational Cell Analytics. And yeah, looking forward to your talk, Constantin. Thanks a lot.
Thanks for the kind introduction, Tishy. Yeah, and so I'll talk a bit about our research related to segment anything and how we adapted it for microscopy today, and also talk a bit more broadly, right, about this effort to build vision foundation models for microscopy. Right, but before we start talking, right, about kind of the topic at hand, right, a bit more what we want to do in my group, right, kind of the vision is to go from images to insight, right, and do this with clinical relevance and in collaboration with life scientists. So this is sort of the motto I came up with at some point. But so what we do in practice is really segmenting and tracking a lot of different types of objects in microscopy. Right here, these are just some examples you see from projects we had here in Göttingen. And then the other focus where I want to switch to a bit is also to do representation learning for microscopy and multimodal data, right, to connect imaging data to genetics or clinical data and so on. And then the last part is also to do protein structure analysis in cryo-ET and what we also do in optical microscopy. Right? So these are sort of things also driven by collaborations here in Göttingen. But yeah, so today in the talk, I want to focus on this part here, right, which is the segmentation and the tracking. And I think, right, all of you are familiar now, right, with the deep learning-based tools for these tasks, right, from UNets as a pretty general architecture to tools like Cellpose and Stardist, right, that already can give us quite good results for a lot of these segmentation tasks, right, if sufficient annotations or ground truths are available, or if the pre-trained models in the case of Cellpose or Stardist sort of work nicely for the data. And luckily, this is quite often the case. So often, right, these tasks are a lot easier than they used to be. But still, right, if these pre-trained models that are available don't really fit the data well, or if we don't have a large annotated similar data set already, then the kind of annotation to get out the ground truth data to train these models really becomes a big bottleneck, right? And this is especially the case for volumetric annotations or also for tracking. And so that's also in our work here, right? We very often still deal with this, that people have like somewhat non-super standard segmentation problems come to us, or at least came to us, and then we would say, okay, give us like 50 to 100 annotated images or 10 to 20 annotated volumes. And I mean, often that's kind of a showstopper for people because either they just don't have the time or expertise to do this, or would often also say, well, then we can just annotate everything ourselves and just don't need to go through this effort, right? So kind of the need that is still there in this domain, right, is to come up with methods that just need either less training data or really generalize better than our existing models in order to speed up these tasks kind of for the everyday segmentation problems or tracking problems that we face in microscopy, right? And so that's where this recent trend in deep learning to build these so-called foundation models, right, which have the goal to solve like a broad set of tasks in a given domain becomes relevant, right? So basically, if we could build such a foundation model for microscopy segmentation or even microscopy more general, right, then we would hope that it could cover a lot more of these kind of bread and butter analysis tasks in segmentation and tracking, and would really speed up the research in this. So this is sort of the whole motivation for the talk today, right? So to look into bit the ideas that we had to build foundation models for microscopy, and of course, specifically segment anything for segmentation, but also talk a bit more about the outlooks on this and so on. Yeah, so this is kind of the background of all of this. And so I dropped this term foundation models a couple of times, right? So what do we actually mean by this, right? So all of this comes from this large language model research, right? So there's the technology underlying ChatGPT and these other kind of powerful online chatbots that probably everyone of you has used in some form or another at this point. And what's underlying these are these big transformer architectures, right? So this is kind of the latest iteration of deep learning architectures used in these that are pre-trained on these massive text data sets, right? So basically to train ChatGPT or similar large language models, right? The companies scrape large parts of text from the internet, and then this is a bit specific to text, right? They use this kind of to do next word prediction or kind of next text prediction and kind of pre-train them this way. Then there's a lot of fine-tuning involved. But in the end, we have this large language model or text foundation model that is helpful in a lot of downstream tasks, right? From being a chatbot to doing machine translation, text analysis, or also assisting in coding. Right? So this is really where this idea about foundation models comes from. And of course, now that this was so successful for text, right, there are also efforts to do the same thing for vision. There are many different examples, just to name a few here, right? So there's this model called CLIP, which connects text and images together, and which at least underlies a lot of the early text to image generation models, right? So things like Midjourney or so on, or DALL-E, right, where you can give a text prompt, like here, an astronaut riding a horse in photorealistic style. These models, which are based on, at least the initial ones that were based on CLIP, would translate this then to an image that matches this description, right? Another thing is segment anything, which we'll talk about in more detail in a second, right? Which basically can solve segmentation tasks in a lot of different settings. And finally, right, so there's also these efforts now to build multimodal model that really combine text and vision in a kind of more combined sense or combined fashion, like GPT-4o, right, where you can also basically ask images about questions like here, right, seeing if it can see that this is an electron microscopy image. Right, so this is in a very, very broad overview, right, the trend behind building vision foundation models. And of course, we don't really have the time to go through all the details there, but just to give you an idea. And of course, training these models, right, is extremely expensive, right? Taking, I don't know, hundreds of, well, I guess tens of millions of dollars at this point to train like the largest scale models. So things that really only large companies can do. But luckily, right, some of these models, like the one we'll talk about in a second, are open source. So we can test them for our use cases and also try to introduce, to improve them for this. This is now where we come to segment anything, right, which is this vision foundation model for segmentation. And before I show how we use this, right, just as a kind of general introduction to this model, right, so kind of the main feature of it, that it can do segmentation from user inputs, which there's also called prompts, right? And so this is what you can see basically in the GIF here, right? So the this point hovering over the image, right? This would be the user input. And then the object highlighted in blue is a segmentation mask predicted by the model, right? And this is what segment anything can do. And it cannot only do this for turtles and frogs or snails, but for a lot of different images, right? Without being necessarily trained for the specific categories or even the imaging domains that are seen in there. And it can also do automatic segmentation, right? In kind of the vanilla version of the model by a fairly naive approach of just covering the image with a grid of points, as you see here, segmenting the object under every point, and then just keeping likely object masks. So this is sort of what was published now about 1.5 years ago by Meta Research with Segment Anything, and really kicked off kind of a lot of research and also applications of this model because of the main features, right? That it had this kind of user-provided segmentation and really worked well in a lot of different domains. So how does this model work in a bit more detail? Right, so we basically have 3 parts to it. So the first part is this big image encoder here. So this is a vision transformer. So as we've seen before, one of these transformer models, but not applied to text, but rather the underlying image. And this is also kind of has the majority of the parameters of the model, and it only sees the input image. And then we have the prompt encoder, right? So this basically takes the user input, right? Like the points, could also be a bounding box, a mask, and the model can also deal with text. And this then is another sort of smaller deep learning model that encodes this. And then finally, there's a mask decoder, right, which takes the features derived from the image and the features derived from these prompts, representation of these prompts, and outputs a mask. And right, all of this is shown here in the sketch for this cat segmentation. Yeah, so this is the basis of this model. So it's actually a fairly straightforward architecture. Just as a bit of a detail, because this will become relevant later, that the model becomes in 3 different sizes, which correspond to different sizes of its image encoder, right? From base, the smallest, to huge, the largest. And now with this, right, we have this model that enables this interactive segmentation. And I mean, this I've already said, right? The really kind of special thing, why this made so many waves, is that it can segment almost arbitrary objects from annotations, right, for many different image modalities based on these user inputs or prompts. And also it's trained in a way that kind of these predictions by the model can be improved, right? Basically, if after putting the first point or just the bounding box around the model, if there's an error, more points can be given in order to improve the prediction. And all of this makes this model very versatile, right? Because one can do the segmentation based on user inputs, but one can also get these prompts from object detectors or from other sources, right? For example, if you have nuclei, you can use these at point annotations to derive seeds for cell segmentation. And the model and the whole kind of interface are open source and fairly well documented, which makes it easy to integrate with other pipelines or extend and so on. Right. So now the question is, right, how did they achieve getting this fairly powerful and generic segmentation model? Right. And there are really 2 core things to this. So the one thing is that it's trained on a very large and diverse data sets with image and object annotations. So this is this segment anything 1B data set, right, with 1 billion masks, so 1 billion annotated objects and 11 million images. And this was annotated in kind of this model in the loop fashion, right? So there were kind of paid human annotators that annotated these images, but already used this interactive segmentation capabilities of the model to speed up this annotation process. Yeah, and having this data set, which is much bigger than any previous segmentation data set, really is at the core of segments anything performance. And then the other thing is the training loop, where the idea is that this basically simulates how a human would interactively annotate an image. And because, right, we deal with microscopy, we already see this here for some EM data, but the same would be the case for kind of a natural image. But the idea is, right, we have some object in our image, right, like here, kind of this part of the neuropil, and we have the ground truth mask, right? So this was annotated beforehand, and we have the precise mask. And then in order to train the model, what is done that we derive a prompt from it, right? Basically an input that a human annotator could make, right? So this means we can either sample a point from this, right? So for example, the centroid of this object, or we compute the bounding box around the object and then gives this to our model as input, right? So basically, let's say we choose the bounding box, we derive it, it goes into the model, and then this is what the model predicts based on this bounding box input. And then based on this, we can compute our loss, right? Basically by comparing the mask here in the ground truth and the predicted mask based on the bounding box, and use this in order to update the parameters of the model. So this is the first step, and we could in principle stop here, but then we wouldn't get these properties of the model to correct segmentations, right? So in order to have this, we actually have further iterations in the training. And for this, we now derive additional prompts to correct the segmentation, right? So you've seen here in this case, right? So the model actually overspilled the segmentation mask here quite a bit, right? So here in this case and in this case, so we can derive an additional prompt to correct this, right? So this basically here is a negative prompt telling the model where this is located, the object isn't. And then we rerun the prediction with this negative prompt. And we see kind of as expected, right? The model now doesn't predict that the neuron is here anymore. However, it makes new mistakes based on this, right? And so then we can derive additional prompts, right? So for the missing prediction in this mitochondria here, and the overspill of the prediction here, again, and we do this by comparing the predictions of the model and the ground truth, right? And keep on correcting this. And basically, we repeat this in each training iteration for a fixed number of times. So I think the exact value is 8, right? And basically do this. And for each of these iteration steps, we accumulate the loss that compares the mask that is true, right? The ground truth mask and the predicted mask. Yeah. And so and then at the end of the training iteration, we average these losses and update the weights as is usually done in deep learning model training. But here, right, instead of this kind of single shot training, image in, segmentation out, compute loss, we kind of simulate how a human would correct the segmentation by deriving these annotations from ground truth. Yeah, so this is really the core, apart from this big data set, that sets segment anything apart and that enables this interactive segmentation capability that we've already seen. Yeah, and so then, right, this is kind of the basic of the segment anything publication. Of course, they evaluate this model for a lot of different settings there and show kind of that it can really work well in many different domains. And based on this, we and also a lot of other groups got sort of excited to test it sort of in their specific niche. And in our case, it's of course microscopy, right? So, and what we basically wanted to check and have checked now is, first of all, how well does SAM work for microscopy data? Then the question is, can we somehow improve it further through microscopy by fine-tuning? And I'll explain this more in a second. And then we've also built this NetPari-based toolkit around it that enables interactive and automatic segmentation, right? And to say, when I say we here, really most of the work was done by my PhD Anway, but also at collaboration with several other members of my group and also the DFKI here in Germany. Yeah, and I mean, I'll talk a bit more about the publication in a second, but right, just to say for the first point, right, so actually in the original SAM paper, they did evaluate the model performance for nucleus segmentation already, right, on this DSB nucleus data set, but it was just like a single and relatively simple segmentation problem. But already this showed that quite some promise for microscopy data. And as you can see here, right, there's really a large variety of imaging domains, right, from natural images to kind of aerial images and so on, that the model was tested on initially. Yeah. One thing, right, before I go to our actual work, just to say, right, so you can find our preprint here. Our actual manuscript is under revision for quite some time, and I hope will be out very soon. And some of the things I'll show today, I unfortunately not yet in the updated version, but I hope they will be available soon. But kind of all the features I show are integrated into our NetPari tool and the rest of the open source software already, so they can be tested or built upon as right now already. Yeah, so what do we actually do in our contribution, right? So the main first thing is just to re-implement this iterative training procedure, right? So this is what I've just shown you at the example of neuron segmentation, right? How given the ground truth segmentation, right, the model is iteratively corrected or learns iteratively to correct segmentation, right? Because while the model itself and kind of the inference code was published by Meta, this code for training was not published, right? So we had to re-implement it, and it's quite a complex procedure, but at some point we got it to work, and then we can use this if we have new segmentation data sets, right? For example, microscopy segmentation, to fine-tune all the SAM components, right? So meaning the image encoder, the one that just processes the image, this prompt encoder for the point or box input, and also mask input, and then the mask decoder that outputs a mask. So this is the first kind of main methodological thing that we did. And the second thing is that we added a new decoder, so a new part to the network for instance segmentation, right? Because as maybe you remember from the beginning, what I showed, that SAM itself, right, it only supports this mode of covering the image with a lot of points to get out an instance segmentation. And as we'll see later, that is quite inefficient. So what we do in addition is to add the segmentation decoder here that regresses distances to the object boundaries and the object center that we can then use to derive segmentation. And I mean, this distance-based segmentation idea is relatively similar to things like this or Self-Host, just a bit simplified. And you can maybe think of this, right, as kind of the decoder of a UNet, but instead of giving it as input this UNet encoder, right, it works on the outputs of the image encoder. And so these are kind of the two kind of main methodological contributions that we did. And then based on this, we wanted to check, right, so now how much can we improve the segment anything model and how well does sort of the default model work? And so for this, we went ahead and basically assembled a large data set from public segmentation data sets, right? So I'll not go through all of this in detail, but we started out with light microscopy, phase contrast images, images from tissue, fluorescence, and so on. And then what we did, we separated these into what we called in-domain data set or our training data set, right? So basically data sets that our model is fine-tuned on, and then unseen data sets, right, that are not used in training at all. And then based on this, right, we compare kind of the vanilla SAM, default SAM, as it was published by Meta for interactive and automatic segmentation, and also compare it to Self-Host just for the automatic instance segmentation part. Yeah, so this is just kind of the first, qualitative results, right, to show you how this works, right? So here, basically, you see the normal SAM model before we fine-tune it for microscopy, and what you see is in blue the prompt, right? So this is kind of corresponding to the user input. In yellow, you see the outline of the true object, and in red is a prediction from the model, right? So and here, for this example, in cell segmentation and phase contrast microscopy, right, you see that the model for this cluster of cells before fine-tuning really doesn't do a good job to find individual cells. After fine-tuning, it does. And I mean, we can see that here for like all of these examples of domains that fine-tuning really improves it quite a bit. And kind of here on the left-hand side, this is what we just called in-domain data sets. So that means that these similar data was part of the training set, right? So these are not training images, these are test images, but it was trained on kind of just the train set of these data sets. That's why we also tested these out-of-domain cases here, right? Where we actually don't use very similar data in training. And we can see now here also that the fine-tuned model does a lot better, right? So we see that it also improves generalization to other microscopy images that are not super similar to the training set. Yeah, so that was kind of the first point. Then to evaluate this more quantitatively, right, we compared the performance for interactive and for automatic segmentation. And so what you see here on the left-hand side is the quality of the default model. And to explain this in a bit more, right, what we do here is we also simulate this iterative correction approach, similar to what you've seen in training, right, where kind of the leftmost bar basically means we either just derive a single point from the ground truth object, and this would be the green part, or we just derive the bounding box from the ground truth object, give it to the model, then segment the mask, and then compare the mask back to the ground truth. And then these further iterations here, that we sample like additional points in order to correct the predictions of the model, right? So this basically simulates what a human would do in practice to correct the model. Right? And so what we see here is that for the default model, we actually don't see a very big improvement by sampling more points, whereas after fine-tuning, right, we overall get a much better performance, and we do get also quite clear improvements with more annotations, basically with the correction. And then also what we have here with these lines is different methods for automatic instance segmentation, right? So the red line here is from SAM, this automatic mask generation. Remember, this is just putting all these points over the images and then deriving objects from this, right? So this does a fairly poor job here for this data set. The line here is what the Self-Host model based on this lifestyle data set. So this is, by the way, phase contrast segmentation. So cell segmentation in phase contrast. So this is what the corresponding Self-Host model does. And then here, this yellow branch is our automatic instance segmentation, right? So the outputs from this decoder kind of post-process to get out instances afterwards. And so this is the result here. So, and basically what we see is that before the fine-tuning, right, sort of the SAM is never really as good as CellPose. And of course, right, comparing these bars to the lines is comparing apples to oranges, because for each of these bars, we have additional information where objects are, because we derive these prompts. So we see, right, that here, even when we indicate each object in the beginning with a bounding box, right, it's not as good as the automatic segmentation from CellPose. Whereas after fine-tuning, we see that CellPose and our automatic segmentation are fairly close to each other, right? So this difference we see here between them is probably not very meaningful. And we see that the automatic segmentation, sorry, the interactive segmentation is actually quite a lot better than all the automatic methods, which is of course what we want, because this has much more information and is closer to what user. Yeah, so this is kind of the first thing where we see that fine-tuning really improves the model by quite a big margin for microscopy data here for a single data set. And importantly, right, this is a training data set. So of course, this is part of a test split, but it has seen very similar data during training. Yeah, one thing just, right, this automatic instance segmentation, as I already said, right, so this is kind of the result from the normal SAM for this automatic mask generation. We see that it's pretty bad, and also on my laptop that it takes quite long. This is afterwards with this more like unit style approach with our additional decoder, where we see that it looks much better and is also much faster. So this is kind of the motivation for doing this. And then, right, I've already talked about this, right? So this is the next part that we then also compare on these, what we call out-of-domain data sets, right? So basically data that was not trained on, and where we also didn't have two similar data sets in training. So we tried on quite a lot of different of those, but here just like 4 of these where we also see pretty consistent improvement of our model, yeah, compared to the default model. And this red line is here the CellPose Cyto2 baseline. And I mean, in general, we see that the automatic segmentation is more or less similar to CellPose. I think if you kind of average out, it's a bit worse than it, but what we get is, of course, this interactive segmentation, yeah, capability in addition to it. So this is kind of the summary of this, right? So we see that fine-tuning on microscopy clearly improves both the interactive and automatic segmentation. This was said already, kind of similar performance to CellPose, I would say a bit worse, but having in addition this interactive segmentation. Yes, so we did the same then for electron microscopy, where we focused on mitochondria segmentation. The overall approach here was fairly similar, just to say that here we profited a lot from this MitoNet publication from Kedar Narayan, who actually went through all this trouble of collecting a large data set from different sources already. So we used this for our training data, and then used additional data sets also from their publication to test the model. And I mean, here you can see this again qualitatively, right? So these are the results before fine-tuning. These are the results after fine-tuning. We again see quite an improvement. And then if we look at the numbers for this, they are quite similar, right? So showing that the model improves clearly for the mito segmentation after fine-tuning. So similar message as to light microscopy. So I will not dwell on this too much, just to say, right, that currently the AM model we do have is fairly specific. So I think we also trained it for nuclei, but it really only works well for mitochondria and for nuclei. So having like a larger diversity from cilia to microvilli or whatever other structures we have in EM. Sorry, so we also have some cilia microvilli in there, but having like other organelles, right, from ER to Golgi, whatever, is currently not in the scope of this model. We are looking into using open organelle data for this, but I'll talk to this later in the outlook. Yeah, okay, this is again just the images, how it looks before and after for the automatic segmentation. Then the next thing what we did, which was also actually also a new kind of methodological contribution, right, is to extend this to 3D segmentation. Right, so this is done fairly naively, basically by segmenting the images slice by slice, right? So basically we either do automatic segmentation or this prompted segmentation for each slice, and then kind of just stitch the segmentation results together, right? So this is what you see here for automatic segmentation, right here for mitochondria segmentation problem, here for cell segmentation problem in Lightsheet. One final thing before we go to the tool and hopefully short live demo if I manage, is then how to further improve this model as a user, right? So because in a nutshell, right, what I showed right now is that we basically tested segment anything for microscopy data. We showed that this fine-tuning improves the model quite a lot, but it's still, right, not a universal segmentation model for microscopy by a long shot, right? So if you have like weird light microscopy domains, it will probably not work that well. In EM, we've just seen, right, that it works only well for mitochondria and nuclei, right? So the goal is to also enable users what we did, right? So basically, if they have a specific segmentation problem that they're interested in, if they can annotate some images, either from some external source or also with the help of SAM's interactive annotation capabilities, right, then they could train kind of their personalized model for this domain. So this is also what we investigated a bit. So how much data is actually needed for this, right? And for this, we used this data set here, basically cell segmentation in immunofluorescence, and looked into how much does the model actually improve when we only provide it with a few images, right? So basically this goes from the initial model here, the default SAM model, and here the our generalist LM model, right? So this is the model we fine-tuned, and going from the initial model that wasn't fine-tuned on this data to fine-tuning images. Yeah, and so unfortunately, it's not super conclusive, right? So for the default model, right, we see quite a nice improvement here in the box-based segmentation performance. Also see some improvement in the interactive segmentation performance. For the generalist model, right, we actually see that we are very good with the boxes here already, but see also here some improvements then with more images. So I think there we really want to do more experiments with more data sets there to make very conclusive statements, but at least we see that based on a few images, right, we can improve the models further, and sort of training this on these is possible on a CPU, but quite painful, right? So it takes 6 hours, and if you have a GPU, it takes 30 minutes. Right? And here are just some how these automatic segmentation results on this data set evolve with the fine-tuning. But yeah, just to say, right, so if you have your own data set, it can be then also further fine-tuned on it, and you can also use our tool that I'll show in a second to annotate for it. Yeah, and so this also brings me then already to how we envision and ourselves use the whole method in practice, right? So basically, if we have new microscopy data, right, we first check how well one of our pre-trained, right, either the LM or EM model works for this, how well the automatic segmentation is, and basically have two options, right? Either the result is already quite good, so then basically we don't need to do anything and can just process the data. But if it's not good enough, that right, then we would use the interactive annotation capabilities of the model to annotate more data, basically to correct these segmentation results. Often, if it's just a small data set, because this correction is quite fast, you are done then, right? And nothing else is really needed. If it's a large data set, right, the idea would be to then retrain the SAM model using kind of this functionality I've just shown, then get your personalized model and kind of repeat this. So basically you get a new automatic segmentation. Hopefully then your result is good enough, but if it's not, you can go on correcting, which is now faster because the model was better for your specific task, and then retrain again. So this is kind of the application in practice. And of course, this is inspired, right, by the human in the loop training that Cellpose already implemented a couple of years ago. The difference is, right, that we have kind of support for more modalities in the beginning, and also that the interactive correction speeds up the annotation process quite a lot. So these are the two advantages. The big disadvantage still is training the model takes quite a bit longer, right? So our process is not really human in the loop unless you have a super powerful GPU, right? It takes a while to do this retraining process. Yeah, so this was kind of all of the background of our work. Then, of course, the one of the main things that we have is the NAPARI tool that makes available the overall segmentation capability of SAM and also integrates our fine-tuned models with it. Right? And just in a nutshell, right, so what we support is 2D segmentation, 3D segmentation using these 3D features that I've shown a couple of slides ago, also tracking, and then, of course, also the fine-tuning on your own data. Yeah, and I mean, it supports both the default and our fine-tuned models, also custom models from users via this functionality, multidimensional segmentation and tracking, and then also tiled predictions for large images. Yeah, just to say, right, so all of this is open source, available. The models are now also on bioimage.io. And yeah, so what I want, rather than showing like images or further details on the plugin, I'll try now to give a live demo of it. So bear with me, right? As usual, things will probably go wrong, especially with Zoom running in the background, but we will give it a try, right? So how you would actually now use this tool for, yeah, some of your data, right? So the tool is implemented as a NAPARI plugin, right? So I've just started NAPARI, and then I can open a sample image. Right? So I'll take one provided by our plugin. And so this is this live cell data. We so phase contrast microscopy segmentation. We work with this quite a lot and so use it often to show it. And right, you can start the plugin here. Right, so basically, if you install the tool, you get this plugin menu in NAPARI, segment anything for microscopy, and then you can select kind of the different tools that we have. Right, so this annotator 2D is the one we'll use now for 2D annotation. Then there's also the one for 3D annotation, the one for tracking, and this image series annotator. So this is basically the same as a 2D annotation tool, but it can run for a large number of images. So basically to go through them and annotate many images fast with the 2D annotation tool. Yeah, so let me start this, right? And so after this, now here our user interface shows up. And so the first thing that we need to do is compute the image embeddings, right? So remember, this is kind of the output of this large vision transformer that is kind of the backbone of segment anything. And here we can also choose the different models, right? So these first ones are the 4 default models of the different sizes. So T is kind of a smaller version published later on. And then come our fine-tuned models, right? So this is the LM, are the models fine-tuned on light microscopy data, come in 3 different sizes. So we didn't fine-tune this huge model because we didn't see much benefits from it when we tested it. And then this here would be the models for electron microscopy. So let's start out with default model, right? So the VITB model, so with this kind of smallest vision transformer. And then when we click compute embeddings, it will compute the image embeddings, right, for our phase contrast image here. And so this is done only once per image, right? So because this takes quite some time. So if now, right, this is on my laptop, so with just a CPU, it takes a couple of seconds. With Zoom running, unfortunately, it will take a couple more seconds because my CPU is quite busy, but we'll see it working here. And now it's done, right? And so this happened now once, right, with this model for this image, and it will not be recomputed until we tell it to. Right, and then we can do the annotation, right? So basically we have this NAPARI layers here in order to provide the inputs to the model, right? So the one option is here to provide these point prompts, right? So this is, I just put this green point here, basically telling the model, okay, here is a cell or an object, and then I can click segmentation and we see that the model does this. And then, right, the way we've implemented this, I can then click commit, basically say, okay, I'm happy with the model, with the object that you found. And then this goes from kind of the current object layer, right? So this was this layer here to the committed objects layer. And this is a bit of logic that you probably won't get with my quick walkthrough here, but this is basically how we keep track of different states of segmented objects, right? And then we could sort of continue with this. We can also, to speed up annotation, click this batched flag here and then annotate multiple cells at a time, right? And basically I can click segment object, it annotates those, I can click commit and say I'm happy with this. Now I'll just deactivate this because right now we'll see the shortcomings of this vanilla or default segment anything model, right? So we see if we click here, it actually segments most of the cell cluster instead of just a single cell. Right, and we can, of course, now try to correct this, right, by providing additional annotations, right? So, for example, we can give the bounding box annotation, right, segment it again, see that this doesn't change that much. We can also give it negative annotations, right? So this means don't segment here, to try to refine the cell. However, as you see, this is quite tedious, actually doesn't give us a very good result, right? So this is now the shortcoming, right, of the default model that just wasn't really trained on microscopy data, so does a fairly poor job when the segmentation problem becomes too difficult. That's why now, right, we switch to our fine-tuned model. So for this, we recompute these embeddings, and it also clears the rest of the state. When this is done, this again takes a couple of seconds. You see it working down here. Yeah, and usually this is faster, but Zoom is quite CPU hungry. Now, yeah, this is done again, right? And so now we can do the same right here. Again, for an easy case, we see that it's the same as before. But now if we go to our more difficult case here, right, we see if we just put a single point, it doesn't actually get the full cell. If we put another point, right, we see that it now finds the full cell here. Right, and I can also do it for this case here. Oh, sorry, wrong button. Segment this, so, and see that it does a fairly decent job, much better than the default model in segmenting these. But of course, by doing this, kind of putting points and correcting it for each cell in the image would still be quite tedious. So we can now also use the automatic segmentation. And again, this now uses kind of this unit style segmentation model that we added to segment anything and uses this for an automatic segmentation. This takes a bit because it now needs to run this additional model, but then we get our segmentation output here. Again, this comes up in a new layer. That's a bit of the technical details, but we see that this gives us a fairly decent result. And of course, now if we wanted to have a really good segmentation, we could go on correcting this. And of course, we can use all the features from the NAPARI layers for this, right? For example, we see this here, right? It's actually one big cell that was split up by our segmentation, but now we can use NAPARI functionality to correct this, right? So we don't have to use SAM for automatic segmentation. We can also use NAPARI, right? For example, we could also remove with this the cells that we've already segmented, like this one and this one, and I think this one. And then at some point, right, when we're happy, can also commit the automatic segmentation. Yeah, and of course, right, we could now also remove parts from the automatic result, correct them with SAM, and so on. Right, so this is in a nutshell how the tool works. Because this is kind of for this phase contrast data, the light microscopy model works for quite a lot of different cell segmentation and also nucleus segmentation tasks. Okay, I think in the interest of time, I will skip the 3D segmentation part because this always works quite poorly when I show it on Zoom. It was too much CPU usage, but just to say, right, so this would work similar where in 3D, where we can also click on objects and then segment one slice and then project the segmentation through the volume and also interactively correct this. Yeah, so this is really as a walkthrough, right, sort of our contribution, our tool as it is right now, and as you can use it, right? So basically, if you go here to our GitHub, you see the software. It's quite extensively documented, including videos, and also, right, if you want to use it, have any problems, any other feedback, that's also, of course, quite welcome. Yeah, but of course, right, we are not really done with this, right, and are still trying to improve the tools and really make it more user-friendly, improve the segmentation performance, right? So the things that we're working on right now is to integrate more efficient training procedures in order to update this user-based fine-tuning, right? So these are kind of parameter-efficient fine-tuning techniques called LoRa, QLoRa. There's a couple of others that we're currently investigating, and these should hopefully enable better training on CPU and also on smaller GPUs. Then the other thing we're still looking into is providing improved models, so also looking into improving the light microscopy model, but especially what we want to do is have a better model for EM that can do more than just mitochondria and nuclei. And for this, at some point, we want to see if we can just pull in the training data published by OpenOrganelle, fine-tune the model on this. And also, I have one student who recently started on building a model specifically for histopathology. So this is kind of the things we're doing right now. And of course, maybe some of you heard this, about months ago or so, also the SAM2, so basically the second version of Segment Anything, was published by Meta, and we are also exploring this for microscopy now. Just to give kind of a brief teaser for this, So the SAM2, the new version is a model now for interactive image and video segmentation, And so its capabilities are shown here in this skit, So basically, instead of just segmenting objects in images, what it can do is now doing this in videos, So you see this here for this soccer player, where the model interactively segments or tracks, the objects like the ball and the player's foot in the video, and where we can also correct errors interactively, here, like the player's foot that was not tracked, So this is kind of now evolution over the initial SAM model that only worked for images. And this is a bit similar to what we already did for the tracking task or the 3D segmentation task in microscopy. But of course, we did this very naively as just kind of this slice-by-slice processing. And in the new model from Meta, this is all integrated in the model already. And in the publication, they also showed some initial results or some kind of proof of concept results for tracking in microscopy here for kind of the cell tracking task. And it cannot deal with dividing objects yet, but at least what they showed worked quite nicely for like a few tracking tasks. So we've started to evaluate it. So I will not spend too long to go over these graphs, but just to say sort of these two bars here, So the blue shaded ones are the original SAM, and the green ones are now, these are two different settings. But so basically just look at the light blue and light green bar. So this is basically original SAM, this is SAM2. And, okay, now I'm confusing myself. Anyway, sorry, just to summarize, what we see is if we read this plot properly, no, I did something in Zoom. Now it's back. Okay. So if we would read this properly, basically our preliminary results tell us that the microscopy image segmentation for SAM2 is not as good as for the original model. And this is now just comparing the performance of the vanilla models without any of our fine-tuning. So basically, if you see, so basically this one here, this light blue, this would be SAM, and this would be SAM2, So we see basically that the strong blue SAM2 is below the light blue SAM mask. And these are similar experiments with interactive corrections as I've seen before. Again, this is quite preliminary because this just came out. But just to say, that it seems like there's still some work needed to take these results we and others had for SAM1 and port it over to SAM2. But just to say, this is kind of one of the next steps to explore how we could also make use then of this better model for especially across time or across the volume for better segmentation tasks. And so then, a bit as an outlook on things we want to do more long-term, is to really incorporate 3D or time properly with the model. Again, this is where SAM2 as a possible backbone is now very attractive, but there are also other ideas to do there. One thing a bit in this more general goal of building these foundation models is also investigating new architecture. So there's this Mamba architecture, which could be a potential replacement for transformers and is much more efficient. And we did some also very preliminary work that shows quite a lot of promise for this. And then also, right, what we currently don't really have is semantic awareness in the segment anything model, right? So basically it just segments all the objects in the image, but it cannot really distinguish between different organelles or different cell types or so on. And this would also be something that's needed in this context to really get a universal segmentation model. And then the final thing that I'm really interested in is also to improve the zero-shot adaptation, or you could also call it example-based segmentation, where, right, you don't need to fine-tune a new model to work better on data, but you just give it kind of example objects that should be segmented. Yeah, so these are the things we, and of course also others, are interested in. But I think in a few years, when all of this is there, when we should have a model that is really much more universal than we have right now for these segmentation tasks. Yeah, that's already everything for my talk, right? So thanks to everyone at Amble who supported me, right? Of course, especially Anna and her group, also Trishy and many others, right? Also people here in Göttingen who are very supportive of what we do. Of course, my group, and especially Anwe, who really was the driving force between all the segment anything work shown today, of course, all the funders and so on. And with this, I'm happy to take any questions.
Constantine, it's Mark. I was wondering, have you tried anything like the skyline approach that Meta showed, where you just have a grid of points and just
Yes.
segment everything under that? Yeah.
Right. So basically, this doesn't really work well in our cases, right? So, So basically, this is this line here, right? What we call AMG, because they call it automatic mass generation, right? So this red line down here, that is this approach. And so we see that for the default model, right, this works quite poorly. After our fine-tuning, right, it goes here, so it improves quite a lot. But the big downside is the efficiency, right? So this is the result before fine-tuning with this model. It takes, on my laptop, 75 seconds. Compared to this, right, our fine-tuned decoder for segmentation takes 9 seconds and is also quite a bit better, right? So this is kind of the downside of this, right? Because what happens is for each point, we have to run the model. And this is quite expensive. And if you have a GPU and are on a cluster or server, right, it doesn't really matter that much. But if you want to run this on a laptop, right, this is really quite painful to wait for 75 seconds to get your segmentation result.
Are the results similar for EM?
It works better for EM if you have things like mitos, right? So that we also had here. Right, so here, I mean, this was the result for mitos. Now again, for the this mask approach, and I mean, it's not perfect, right? But it's much better in actually finding mitochondria than it was before to find cells. But the inefficiency, of course, stays.
Yeah. Got it. Thank you.
All right. I have a question. In this initial video, actually, with this frog, like it looked like that I mean, sometimes you can have one big object, let's say a cell, but it might have a nucleus inside. So there it looked like that if you actually zoom into the image and then click again, it does something else than if you're zoomed out. Is that how it works, or?
No, not really. So the idea here is it always shows you what's hovered under it, and then it puts a negative point, right?
It's a
Which
negative
we've also
point.
seen in the video, right? To separate the frog from the snail. So that's what's happening here.
So I cannot just click on the snail. That would not work here, probably.
I mean, it really, right? It's kind of, there's this ambiguity a bit of what is an object, and
Yeah,
you can only
exactly.
resolve this by multiple prompts.
Okay, because zooming it would also be in principle a prompt, right? That you say, hey, you know what I mean?
Not quite, but I'm just, right, in this case, you could just do the reverse, right? You could basically put a positive prompt on the snail and the negative prompt on the frog, and you would get out the snail.
Okay, thanks. Anne, you have a question?
Yeah, so you have these two encoders, one of which is really hard to retrain, where how fast as a user do you know, oh, the issue is actually in the image encoding and I will not be able to get a good enough prompt out of by just extra training? Because how fast does this satisfy, how fast is it trainable, the prompt encoding? Because otherwise you don't know, essentially.
So, I mean, that's a good question. Just first, to say what we do now, we always update the full model in the current setting, because we found if you only update the prompt encoder or only the mask decoder, the gains are quite small, right? So basically, I mean, it gives you some gains, but they're really not worth it. So we always update the full thing, which is also why I showed these numbers briefly on the CPU. That's why it takes so long to do there. So I would say right now, I think you can always, well, this is a strong statement, but most of the time improve it through fine-tuning, but it will just take its time for it, especially if you don't have a powerful GPU. That's kind of the idea behind these parameter-efficient fine-tuning techniques that I mentioned in the end, right? So there, instead of fine-tuning this full image encoder, you only fine-tune small parts or extra parameters. And that's how we hope to get this more efficient, but it's kind of still ongoing research.
Thanks.
Maybe again, like, so fluorescent 3D nuclei fully automatic. Did you try this?
Yes.
And
it works decently. It depends on how big the nuclei are, right? So if they're very small, it's a bit tricky. If they're big enough, it works quite well. But I mean, it's part of these, right? We have really a lot of data sets, and I mean, I always struggle a bit to show this, but so yeah, this here actually, right, this is what we call here mouse embryo data set. These are 3D fluorescent nuclei. And we have
okay,
a few
so that's worth trying. And then in Apari, I would just load the 3D image and say automatic or something.
Yes.
Okay. Yeah. Cool.
I guess I have a question. Hi, Constantine. This is John.
Hi,
nice
John.
talk.
Hi. Thanks.
I was curious. So I guess in our experience when we were trying this, it seems to prefer blobs and doesn't really like long and thin things like microtubules, say. So I guess, I mean, some of the data in the live cell, ooh, nice, I get a video, hi. The cells in the live cell that you showed during the demo have some kind of long stringy things and are not super convex, but I guess I was curious if which data sets were the least blob-like and how much fine-tuning helped on things like that.
Yeah, so I think, right, so live cell really has some non-convex things. Also, these debugs bacteria has like a lot quite stringy objects. I think if it's just kind of a single stringy object, it can actually work quite nicely, especially after fine-tuning on it. If you have like more complicated, right, sort of, right, like a network of fibers or strings, something like this, then I think even fine-tuning won't really work well, especially if you have 3D structures and so on. So that's kind of our experience, right? So anything like vasculature, segmentation, and so on, I think it's just not a very good model for it. But if it's really just like single rod-like or more complex shaped objects, it can do fine. That's at least my experience with it. But I think it needs like some fine-tuning on the specific data to work really well.
Nice. Yeah, thanks.
Any other questions? Maybe I have a final one then. So in general, I had the feeling box works better than point annotation.
Yes.
Okay.
Yeah.
Good.
Yeah. I mean, it also just provides more
makes sense. Yeah.
More information, right?
But
the idea really is with this correction that you can combine them, and of course, this kind of correction can only be done by points, right? You can only provide one box, but you can always add more points to better define the object.
And in your plugin, as long as I don't click new or something, everything I click will be used for the same. So you can mix, you can match, you can do both point and box for the same object.
You can mix them as you want.
Okay, cool.